Welcome back to the third episode of Overview Biostatistics by Dr. Pandit. We continue this flowchart that previously we have worked with the components of statistics due to that there is variations of the data being collected and we need to quantify it. Now, another one that is very important and statistics can help in some way as well is the bias. So I will be back to this. But I emphasize again that the role of statistics is to quantify errors for judgment. And there are two components of statistics for that purpose. The first one is parameter estimations. We reported it as the confidence interval. The most common reporting style is put the probability in there, like 95% confidence interval. And the second one is the p-value from hypothesis testing. That's quantify errors for judgment. Okay. So another component of the noise is about the bias. The first one could occur here. That means how we draw a sample from the populations can introduce the first group of bias called selection bias. If the sample being selected purposively, that means we get everything here wrongly because it's not representative of the populations. That means the data being collected from this bias sample will bias whatever statistical method being analyzed, also wrong because quantify the wrong thing. So bias that make the noise large, the first one is selection bias occur in here. So that is why we really need to describe in details, allow being replicable by others. In the method sections of the protocol, the research protocol, or in the manuscript or the paper submit for publications, we really need to describe in detail how the sample being selected. In an observational study, we call it sample selection. Sometimes we call random sampling. How the sample being selected randomly or purposely. In experimental study, like randomized control trial, we call this section random allocations, not random sampling, random allocation. Allocate the sample to groups. So that's random allocation or randomization. In either case, we really need details so that the audience can judge if there are bias here. Sometimes it's not due to that selections. It could occur due to like dropout, lost to follow up, refused to participate. The groups being available for the analysis are selected by some mechanism. This also cause bias. So that is why we really need some flow chart about some flow diagram about the study design from the populations, how many dropouts, things like that. Okay. The second bias, the group of bias, actually the bias has many, there are more than 100 names. So I group here only 3. This is the second one. The second one is called information bias. It's occur where the data being collected is not correct systematically. Okay. Means what? Means, for example, if the group, let's say the vaccination example, if the vaccinated group being known by the assessor that these participants are in vaccinated group, they may try to measure carefully about the infection, for example, in the vaccinated group. If the assessor of the outcome knows that group belongs to the control groups, if that participant is infected, they may dig further. If not, they may go ahead, for example. So here, make the information being collected in the forms wrongly, systematically. One group tend to be more in another group. But if the two groups being measured the same way, it can error, but can be just randomly, no bias. Okay, that is what I mean by random error or bias. Bias is systematic error. The same way with observational study like case control study. Usually case control study, people blame it being sensitive to recall bias. Okay, it's not the case. It is not for every cases, because if that recall could occur randomly, not just in one particular group, there is no bias. So make sure you understand about bias. It's a systematic error. In this component, we really need to put in details of method sections about how the data being collected, outcome and outcome measurements is important in the protocol. Statistics also need to understand about this basis, because it's will determine statistical method to be used. This is the second component. And the last one is called confounding bias. That means there are some independent variables that is not the main interest, or sometimes we call the covariates, play roles or distort the findings by this. For example, if the vaccinated group in that paper, if the vaccinated group compared with the the control group, which is not vaccinated, they get exposed or vaccinated and not vaccinated. But there are some variables that is related to HIV, for example. There are more tendency to get infected. For example, they have lower immunity in one group as compared to another group. So it's not because of the vaccine, but it's because of another factors. This is called confounder. So if being collected, the confounder being collected, statistics can play roles here. We will describe in the method sections how statistics account for these factors, I mean effect of the other factors or confounding bias. Okay, so these are three groups of biases that could distort the findings and make an invalid conclusion about this component, the bias. Statistics play roles a lot here about controlling for confounding bias using statistical modeling. Statistics can play roles here, information bias, by many approaches to see if there is information bias here, or even the selection bias. For example, statistics could explore if the two groups is balanced or similar in the baseline characteristics. So these are another area where statistics play roles. It's not just quantify error for judgment, but do some analysis based on a known understanding about biases, explore it, investigate it, demonstrate it, that there is or there is not the case in that study. Sometimes we call sensitivity analysis. Put it in the discussion section so that we ensure the conclusion is valid. The three components of biases or systematic error here is well handling by study design and well conducted research. So that is why a good research really need to have a very good documentations about all this and how it being conducted, how these confounding, these biases could be prevented or minimized. Details need to be available in the protocol or also in the full articles. So, selection bias, information bias, and confounding bias can be handled by statistics if the data is available. Like selection bias and information bias can be assessed and make a caution if they present. A confounding bias can be adjusted for it using the multivariable analysis or statistical modeling. If not, we mislead the society. its danger. In research, some say that useless is better than misleading. See you the next episode.